Recording in progress.
Welcome to Progenesis Academy webinars. Today we're going to have a very exciting topic. Our speaker is Dr. Fadi Sharara. He is the medical director and founder of Virginia Center for Reproductive Medicine. He's also the clinical professor at the Department of OBGYN, George Washington University. Thank you so much for joining us.
Thank you for having me. Great.
Awesome.
Thank you, Nabil, for the kind introduction. First, happy 2/2/22 for everyone who's attending tonight, and happy Year of the Tiger if you are into the Chinese calendar. So I was tasked with presenting something I think is very exciting, which is talking about the prospect of metabolomic profiling in preimplantation genetic testing. And as can we have the next slide, Nabil? Can I move them, or you have to move everyone? Here we go. Okay, so we all know that IVF success has recently plateaued. For those of us who have been at least reading about IVF, and for those of us who have gray hair, the advances in ART have been absolutely breathtaking for anyone who witnessed the early days of IVF. Live birth rates with single embryo transfer and PGTA have exceeded 60% in many programs now. However, there has not been any recent breakthroughs allowing success rates to make the next leap. Are we at the point of maximum success, or is there room for another increase? And there has been, over the past few years, search for non-invasive testing to increase ART outcomes. So we recently published this paper, and hopefully Dr. Arash can put a link to this paper so that everybody will have it. We published it in March of this year, and I'm going to read the abstract because I really think this is very important to what we're going to talk about. So the title is Current Advancement in Non-Invasive Profiling of the Embryo Culture Media Secretome. And there has been over 8 million babies born through IVF, and this number continues to grow. There is a global trend to perform elective single embryo transfers, avoiding risk associated with multiple pregnancies, of course. It's therefore important to understand where current research of non-invasive testing for embryos stands, and what are the most promising techniques currently used. Furthermore, it's important to identify the potential to translate research and development into clinically applicable methods that ultimately improve live birth and reduce time to pregnancy. The current focus in the field of human reproductive medicine is to develop a more rapid, quantitative, and non-invasive test. Some of the most promising fields of research for non-invasive assays comprise cell-free DNA analysis, microscopy techniques coupled with AI, and omics analysis of the spent blastocyst media. High-throughput proteomics and metabolomics technologies are valuable tools for non-invasive embryo analysis. The biggest advantages of such technology are that it can differentiate between the embryos that appear morphologically identical and has the potential to identify the ploidy status non-invasively prior to transfer in fresh cycle or before vitrification for a later frozen embryo transfer. The biggest word in this is differentiate between embryos that appear morphologically identical, even if they're PGA tested. We all have embryos that are AAs or ABs, and they look exactly the same. Which one would you transfer for that patient? All right, next slide, please. All right, so this is a table from that paper, and many of you know some of these things, but I will assure you some of you have never heard of some of these technologies that are being used right now. Under microscopies, I'm not going to spend much time. We all know about time-lapse, polarization microscopes, but under spent media analysis, there's RECGH, there's NGS, there's obviously cell-free DNA genetic testing by PCR or NGS, electrochemical impedance spectroscopy. That's one thing. Enzyme ELISA, HPLCs, liquid chromatography, gas chromatography, The one I'm going to spend some time on is what's called the Maldi TOF, which is matrix-assisted laser desorption ionization time of flight mass spectroscopy. It's a long word, but it's a very exciting new technology. There's also obviously microarrays, microfluidics, nuclear magnetic resonance spectroscopy, proximity extension assays, respiratometry. So this looking at basically respiration of these embryos in culture. Thermochemiluminescence, ultramicrofluorescence, and vibration spectroscopy. So these, and the references are there. All these technologies are things that are being evaluated right now. Next slide, please. So the slide before that, please. So when we do spent media analysis, what do we take a few microliters, as little as 1 microliter in many instances when we're using a Maldi, and we put this obviously in an Eppendorf tube, we freeze it, or we can run many things on it: genome analysis, transcriptome analysis, proteome, and metabolome. And under genome, you can look at coding genes, you can look at non-coding genes. Transcriptome, obviously that there are mRNAs, small non-coding RNAs, and long non-coding RNAs. Under proteome, you look at structural proteins, non-structural proteins, functional proteins, and enzymes. And under metabolomes, you look at lipids, amino acids, carbohydrates, and nucleotides. Next slide, please. So what's one of the technologies I mentioned is the Maldi. What do we do with the Maldi? You get the sample from the blastocyst media, and you put it on a slide that basically runs a plate that runs up to 320 samples at once, and it gives us very high throughput data acquisition in terms of spectra. And then you make an algorithm based on outcomes and these spectra, and you, from using these algorithms, you can develop basically a spectral data analyzed by 2 optimized algorithms, which gives you multiple things to look at. You can look at euploidy or aneuploidy and give these embryos an individual score for each embryo. So you have embryos that don't look as great, don't give us a nice spectra, and these are embryos that, even if they're genetically normal, would give us the lowest chance at these embryos implanting. And we can go up to 96% in terms of giving a score on embryos, and as low as 14%. So you get, if you have 4 embryos that are euploid, you get basically a score for each single one, and then you would decide which embryo you're going to transfer. Next slide, please. So this is what I'm going to introduce the Maldi. Next slide, please. So why do we need to use these technologies? And this is, again, a quick reintroduction of the topic. It's non-invasive sampling. You take basically media from the media that the embryo is growing in. We use minimal volume. So in the Maldi, you can use as low as 1 microliters, and you can even dilute it further, because the Maldi can detect concentrations in femtomoles, so in incredibly small quantities. And it's a very rapid analysis. It takes about 20 minutes to prepare, and the run is 5 minutes. Next slide, please. And as we mentioned earlier, this is a nice slide just looking at an analysis of metabolome. You can analyze protein and secretome, and you can also look at exosome. And I will talk about exosome a little bit moving down in the presentation. Another slide, please. Next slide. So this is the Maldi. The original Maldi, I've seen these, and they were as big as basically a fridge. And now these are benchtop Maldis, and they have reduced significantly the laser path that you'd have to get to basically read the spectra. And this can be put in any IVF lab. So it's a benchtop at this point. You can do the analysis. If you're doing a fresh embryo transfer, you can do the analysis right before you have 4 or 5 embryos, you want to transfer a single embryo, literally within 25 minutes, you can run the analysis and decide which embryo you're going to transfer. And if you're doing frozen embryo transfers, you can collect as up to 320 samples, as I said, and you can run them all at the same time. So you can run multiple patients, multiple embryos from each patient, up to 320. So every 25 minutes, literally, you can run up to 320 samples in this machine. Next slide, please. So what has been published to date, and what recent data does the company has? Next slide, please. So I'm going to review 2 papers that we published. The first one we published in 2019, which was the initial paper that we did. It was based on 401 samples of spent culture media, and of which 136 were used to construct a predictive model. The media sample, you can get them, you can freeze them at -20, and you can store them as long as you want. And you just thaw them before, you get 1 microliter of the spent media, and you run them in the Maldi. And the mass range is between actually as low as 200 up to 17,000. But this one, we looked at 2,000 to 17,000. And then we devised a specific algorithm based on these results. Next slide, please. The next paper we published in 2020, we tried to differentiate if we could see differences between euploid embryos versus aneuploid embryos in non-invasive way. So in this study, we had 292 samples. Of these, 149 were euploid and 165 were aneuploid. And they were all tested by PGTAs. And we looked at the secretome mass spent media using the Maldi TOF. And what we found, that there were distinct spectral patterns between euploid and aneuploid genotypes in embryo culture media. There were 12 characteristic peak signatures for euploid embryos and 17 for aneuploid embryos. And data analysis also revealed a high degree of complementarity, among regions, showing that 22 regions that required to differentiate between genotypes with sensitivity of 84% and the false predictive rate of 18%. And if we can look at the next slide, please, I will show you what I'm talking about. So these are the different peak signatures that we see. And you can see euploid being in blue and red are the aneuploid embryos. And you plot them on this to see where they are. And if we look at the next slide, please, and you do these in a, you represent them basically across a number of regions. These are the 12 regions we looked at. And you see that there were marked differences between euploid embryos and aneuploid embryos. And that's based, as I said, on 292 different samples. Next slide, please. So what else do we have? What do we have after 5 years of collecting data? We have more than 3,000 embryos, embryo culture media that we collected. And we have, when you change it of embryologists and protocol, we removed 1,194 samples, no outcome data on 952. That leaves us with 1,102 data files. We remove inconsistent labeling or miscarriages, aneuploidies, biochemical pregnancies, ectopic or blighted ovum. And we looked at 604 data files now. We removed twins and triplets, 1 case of triplet, and we have 570 data files that we have right now that we are identifying. And we have noticed that hatching is very, very critical for this. Unhatched embryos do not secrete much into the spent media. And because most transfers are now in FETs, everybody is hatching these embryos before doing a transfer. And all you need is basically 30 minutes of this embryo being in the spent media before doing a transfer, and you will get an accurate read on this embryo. Next slide, please. So of these 570 spectral data, 203 were fresh, 366 were frozen, and we have implanted and non-implanted and hatched and non-hatched. Next slide, please. And this is what we found. So when we put the whole sample, 570 samples, and we looked at the spectra, there were definitely clear differences. In the spectra between 500 and 600 megahertz, there were differences between those that were positive or negative. And positive here, I mean live birth. Negative meaning no implantation. And down between 700 to 800, there were clear differences between, again, live birth or sustained implantation and negative embryos that did not lead to a pregnancy. So you can clearly, just by looking at the spectra, you can find out that there were differences between those embryos that implanted and those that did not implant. Next slide, please. So we just started a large prospective study at a leading ART program in the US, evaluating fresh and frozen samples of single embryo transfers with profiles analyzed in real time. All the blastocysts have undergone PGTA. The best scores, which is the score for the embryos that we have, will be applied prospectively before frozen embryo transfers and the outcomes recorded. And we hope that we will have results to present at ASRM. Next slide, please. What else can the Maldi do other than just looking at spent media? Next slide, please. Next slide, please, Nabil. There we go. Okay, so I'm going to touch a little bit about something called exosomes. So exosomes are cell-secreted membrane-derived vesicles that provide new dimensions for the concept of intercellular signaling. And if you look at picture C, you see that these exosomes are very critical in embryo endometrium crosstalk. When we talk to patients about, putting a euploid embryo, and, I give them the analogy of a spaceship that needs to dock. So they check each other beforehand, make sure that this is not an enemy craft, and then so this mutual crosstalk happens by the use of exosome. And these exosomes are really critical to allow an embryo to implant into the endometrium. So they don't contain just proteins. They also contain messenger RNAs, microRNAs, double-stranded or genomic DNAs. The molecular cargoes carried by exosome affect cellular activity via ready-made proteins and microRNAs, or by translation of transferred mRNAs. So these exosomes are really very critical in implantation. And this is something that the Maldi can detect. Next slide, please. What else can the Maldi detect? MicroRNAs, of course, which are small, on average, only 22 nucleotides long. They're non-coding RNAs that regulate gene expression and have been implicated in a wide array of biological processes, including early embryo development and stem cell differentiation. Blastocysts express microRNAs, which may be important for their survival. Therefore, expression of microRNAs in exosomes isolated in vitro culture media could be the basis for the development of novel biological markers for selection of better quality embryos and for subsequent successful pregnancy. And if you can see from the picture A and B under these microRNAs, they can be sequenced even by the Maldi, by selective cleavage. So there's a lot of things that we can use other than just looking at metabolomes. Next slide, please. So what else can the Maldi does? There's something very exciting called mass spec imaging, and you can profile oocytes and you can profile blastocysts. So you can look at these as a mass. You get a mass spec generated image, and you can look at blastocysts, you can look at oocytes, and probably you can look at even at endometrial endometrium. Next slide, please. And this is, for example, looking at an image of an oocyte, and you can see these very clearly. You can get those in using the Maldi and see a different profile than you would get by just looking at these oocytes or blastocysts. Next slide, please. So non-invasive methods are being developed by multiple investigators now and will be used further to improve ART success. And I personally believe that days of possibly 85 to 90% success rates are not far away if we can add to what we know already from just PGTA and looking at methods that can not just complement, but you can use them by themselves in a setting where people cannot afford to do PGTA testing. Next slide, please. And thank you very much for your attention. And I'll let Dr. Arash take over from here, and I'll be more than happy to answer any questions. Thank you.
Awesome. Thank you so much. This was really an interesting presentation. I have seen so many details that I was not aware of when it comes to the Maldi top and all the different methodology. So my question to you, I'm going to start from the end, and then we'll go back to the beginning of the story. The current accuracy of the Maldi TOF using your papers, it's about 84. Is that right?
84% with the last paper.
84%.
And we're hoping we can improve this further.
Yeah, so that's similar to spent media. Spent media maximum correlation we have seen so far is about 84%. Do you see any synergy between the two where you can look both at genomics and also at metabolomic?
Yeah, excellent question. I think ultimately, yes. You know, if you're doing PGTA and not looking at anything else, and you transfer a single embryo, we're maxing out at possibly 65, 70%, let's just say. So when you have the spent media, you can do Maldi, you can do non-invasive PGT testing, anything you can use. Now, if you're getting 84%, some other people are not having that much of a success with using PGTA on spent media. You know, other investigators have published before, they get a better read on day 6 or day 7, and obviously changing media on day 4. This is obviously something that is very cumbersome for the lab, and I don't think many people are willing to do that. No one's going to wait until day 6 or 7 for their embryos before they start biopsying them or do something like this. So I think non-invasive testing is still in its infancy, but I really believe the more people do testing and the more people look at different techniques, as I showed from that table, we're going to get to a point where we will have something that is additional to what we have right now.
Yeah, and that's very excellent point. The other question is the size of the molecules. Obviously, you have been looking at small molecules to very large molecules. And it depends if you're looking at transcriptomes or some protein mix, or if you're looking at macromolecule. Are they all the same? They have the same potential as far as improving the correlation, or do you see a target that is more interesting than another?
Excellent. So right now, I cannot tell you more than what I just presented, but there will be small things, including, for example, microRNAs and exosomes that may end up being more important than macromolecules.
Yeah, I have had some talk with people from mass spec, and they tend to like more small range. They say that it is better control when you try to tight the conditions of measurements. So when you have a spec media sample, if you change the media, you will change that distribution, right?
Correct.
So you have to stay with the same media, same condition at all time.
Right, which is why this is going to be sort of program specific. So every program uses different media, okay? And it all depends how much you equilibrate your embryo before you do the transfer. But all we're saying is with the Maldi, 30 minutes equilibration of the embryo after you hatch before you do the transfer is enough to have a lot of substances that are in the spent media already. But there may be, with different media, we may get maybe slightly different results. The papers we published are basically on what we've been doing, and we have changed media over the past 3 years, so we can look at before and after. Maybe media will not be different. It's as long as people hatch the same way and basically culture the same way. So maybe there won't be any differences with different media.
Yeah, that's excellent. I can definitely see a situation where each lab has to have different validation.
Yes.
And there will be no universal criteria for measurements, because you're going to have to subtract the background, and that background or noise is coming from different media type and different culturing condition and all that, right? Do you think that would be the case, or?
So maybe not. So what we're doing right now, in the new study that we're doing, we're looking at a different program doing exactly the same thing. Okay. And we have to see, with the data that we saw in the media that we sent from my lab, look any different from other lab. The very few spectra that we've seen so far from the new lab suggest that there are no differences.
No differences.
Right.
That's awesome. I'm going to jump into a question from the audience, and then I'll come back to the discussion. So we have a question. It says, do you think these technologies may or might be used to optimize culture media or to provide QA for a lab for culturing systems? So not just beside the the diagnostics, actually just the QC and how the culture media should be.
Look, that's an excellent question. We're assuming everybody is running an excellent lab, okay, and they know how to grow blastocysts and all these things. It's too soon. I'm not so sure this is going to be as a QC, but if you're getting the spectra every single time and they correlate, then you must have fantastic QC in your lab. We always run these samples in duplicates, okay, always. We want to make sure that these two spectra from the same patient always show exactly the same thing. So just like any experiment, we run everything in duplicate. There's always a blank when we do this assay. So you want to make sure that you're not reading something that is not there.
Yeah. And I think the question is not about diagnostics or choosing the right embryo. It's about other aspects of optimization. How,
right.
When you're culturing the embryos, you don't have any control of what is there, right? You just use the media and you don't know the changes that happens in real time. And so potentially this could also be used to kind of QC the lab, the culture media real time.
Too soon to tell. I think we're just really at the infancy of this field. The more data points we have, the more things we can look at. And the more labs that are doing the same thing, the more we can correlate between different programs. If we're all showing the same thing, then we're doing things the same way. And that you have, at that point, what you're doing in terms of QC is really working. The more data points we have, we'd love to have hundreds of thousands of embryos to look at from many different programs, just to make sure that they correlate. With PGTA now, we don't even have correlation among different labs, the genetics labs. Yeah. So we hope with this one, we would have correlation among different labs, because everybody is going to run the same thing. I highly suspect the media may not make much of a difference. It's just growing to day 5 or day 6 and seeing. But there could be differences among different programs. That's why it's really critical for different programs to collect their own data. And the algorithm that we did was specifically based on my clinic. If some clinics, we may have to adjust the algorithm a little bit differently based on what outcomes we're getting from your lab. Yeah.
And you mentioned earlier the differences between hatching and no hatching,
right,
fresh and frozen. So when it comes to hatching, does it provide any benefit as far as collection method?
Yes, very much so, because these are very small molecules, okay, especially like exosomes and all the secretomes that we have, and the zona pellucida is quite thick. So when you do hatching, I mean, we looked at hatching versus non-hatching, and hatching embryos have tons of metabolites at that point compared to non-hatch samples. So to really get a good read, you have to hatch, which we all do at this point for frozen transfers.
Yeah, I believe in the spec media research that we have seen so far, hatching provide about 5%, an added 5% in the correlation between media and embryo. So it does help in some ways, for sure.
Yeah,
But
for the Maldi, again, because the Maldi discovers things that are in femtomoles, okay, which is, you know, minuscule amounts. We saw huge differences between hatched and non-hatched embryos.
Yeah, and the spent media, not the spent media, but the actual embryo fluid that you extract has the potential to have more metabolites, maybe better metabolites than the one that are in the exterior, just because the zona does provide some sort of isolation to the embryo. I looked at the details of the mass spec that you provided. So I've seen the two peaks, the 560, 540, and then the range 560 to 760 to 800. Have you guys figured out what components, what product is that?
Yes. So we're still looking. There's a lot, but they're definitely metabolites that you would think would be very important for embryo health. Riboflavin, for example, is one of them. There are a lot of other things. So wait on this. We'll get more data. But yes, all these substances are actually critical for embryo health.
But it looks like they are small molecules just because of the mass, right? They cannot be macromolecules. They have to
correct.
be.
They're all small molecules. Absolutely.
Molecules. Yeah. Very interesting. What is the you know, at the beginning of your presentation, you showed us the Maldov latest equipment, which is a benchtop, a very nice benchtop, very small. I think it's very affordable to have it in the lab. I would buy it.
I wouldn't say affordable. It's about $150.
I will buy it.
Yeah, it's about $150,000.
Yeah. Well, all equipment in our laboratories are the $150,000.
Oh, absolutely. Yeah, you know, you know, the great thing, Nabil, if you have access to cement media, you have to get a Maldi. It's an unbelievable machine. You know, this is the picture I showed you is a Shimadzu 8020, which is you know, one of the most commonly used Maldis, and it's just an amazing machine.
Yeah. So back to that question, do you see, is this a direction that would be where end users, IVF centers will have more access to than the traditional, you know, genomics and genetics?
Yeah, I definitely do. I mean, you know, this is the, I mean, you know, 150 in a big lab is definitely something that's affordable. Okay. I mean, in a small clinic, it doesn't make much sense. It all depends on the volume. And you can have one central Maldi and have many clinics send samples to, because it takes 25 minutes to really run the sample. So it's very powerful as a machine. And as I said, if you are doing fresh transfers, you bring the patient in, you run the Maldi, you get an answer in 25 minutes, and you put embryo B rather than embryo A, for example. And the patient gets pregnant. So you can use this information in a prospective way. But in terms of where the field is going, there is no question that the field is going to go towards looking at, I mean, we've done the genomics, okay? So we're doing PGTA. We have, what else can we look at? We've done ERAs, if you believe in ERAs and endometrial receptivity and all these type of things. And yet we're all stuck at the 60–70% range. We're not breaking this, even in young women. And if you use ERAs and you say, okay, you believe in it, then the endometrial factor, you at least have minimized as much as possible. And Dr. Scott's nice paper about the, basically there's the pregnancy rate after 3 successful single embryo transfers reaching close to about 92% is amazing. But we want to get to 90% from the first transfer, not after 3 transfers. Hence why I believe everybody is going to start looking at these technologies.
Yeah, but also the implantation rate is not just limited to the potential,
not just the embryo.
Just the embryo. So
right.
I think it potentially could improve the implantation rate, but we're going to still face that limitation of the transfer technique and
sure. I'm assuming you optimize these. Let's say you have somebody who's done tens of thousands of transfers and can do transfer in their sleep, and you have an ideal patient, for example, not somebody with fibroids, not somebody with adenomyosis, not somebody with a unicornuate uterus or whatever. So in a normal patient, let's say normal, younger than 42, let's just say, or younger than 40 if you want, and you have a euploid embryo, what we're trying to find out is, and you have 3 embryos that are euploids, are there, and they all look AA or AB, are there differences among these embryos? And the short answer is absolutely there are. Okay, so if you can prioritize and basically make a rank for these embryos, which one to be transferred, if you have euploid embryos and you run these tests and you identify which embryo has the highest chance at implanting, that's the embryo you want to transfer from the first time.
Absolutely. So back to that crosstalk between the embryo and the endometrium. You have put a really nice slide where you talked about exosomes and the components of the exosome, everything that is channeled with the exosome, the proteins, the amino acid, the microRNA, the regular messenger RNA. How Maldov can help? I guess you're going to have to figure out what happens in the other side too, right? Not just the embryo. How do you see this, like from a practical?
Excellent question. So ultimately, you're going to also have to look at not just the spent media, but also the endometrial interface.
Yeah.
So stay tuned. We're talking about this actually today.
Oh, awesome. Are you presenting?
Not presenting. Hopefully, we will have something down the road.
Yeah. Okay. So eventually, we may know about an assay that looks at a more sophisticated era assays.
Right.
This time looking for exosomes.
Absolutely. Absolutely. Because even you do an ERA, and if you read the ERA papers, you increase the chance of a live birth from 50-some% up to about 70-71%. We want to maximize this beyond that point. So your question is absolutely excellent. We're looking at the spent media and the embryo. Okay, the endometrium is still quite critical.
Yeah,
okay?
Absolutely.
Now with new, with looking at the endometrium much more, we have decreased the incidence of problems with the endometrium, especially with ERAs, down to maybe about, let's say, 10%. Okay, but if we can have a test that we can look at which endometrium is receptive in not just in a macro way, looking at the genomes, but also look at metabolomes for this, which is one of the companies, iGenomics has been looking at this. But hopefully we would like to have a test that's even more accurate than this.
Yeah, and if you're trying to get a look at the endometrium from a metabolic perspective, you need some time, right? You cannot do, this is not a fresh transfer type of situation, right? You're gonna need to, it may take a little bit of time, no?
It will. I mean, you can, in the early days of, noise, or, when Bruce Lessey looked at the integrins or whatever, you don't need hundreds of patients to try to find out what, you can bring somebody and start progesterone and look at day 1, day 2, day 3, day 4, day 5, day 6, day 7, and find out what type of metabolomes are being different between those. I'm not aware of this. Other people may have looked at that, but I haven't read that literature lately.
That's so exciting. So the metabolic activity of the embryo is definitely, I mean, my opinion is the way to go. Besides genomics will give you an idea, and sometimes they don't overlap. I don't think they overlap 100%. I think there are situations where you're going to see a metabolically active embryo, good candidate for transfer, but they may have a trisomy or monosomy, and therefore may fail to implant. And on the other side, you may find a very beautiful embryo on a grade scale that is, according to genomics, is euploid, but not metabolically fit for transfers. I think there is an overlap there, but there's not a 100% overlap. What do you think?
Absolutely. That's an excellent point. In the very early days of PGA testing, and you recall that very well, we were told, look, an embryo, if it's euploid, then morphology is not going to matter. That's absolutely not true. Morphology still matters. Okay, so a beautiful embryo has a higher chance to implant than a BC embryo that happens to be euploid. It's exactly the same story from a metabolic point of view. It's probably even more important. You may have a BB embryo that is more metabolically, let's say, normal than an AA blastocyst that is euploid but not as metabolically active.
Exactly. I'm going to go ahead and share the paper that you mentioned. I don't know how to share this, but I think I can just maybe copy.
Yeah, if anybody is interested in this paper, please reach out to Dr. Raj or your Progenesis rep, and they can definitely email you this.
Yeah, and I just copy paste the actual title in the chat form. So if you guys are interested in looking at this paper, look at the active chat and you can copy paste.
I personally believe this is really the future. We have, first we were stuck with morphology. We're doing day 3, then we were doing blasts, and now we're doing PGTA tested embryos. We're just maxing out. I have personally not seen anything over the past, 4 or 5 years that have led us to a higher chance of a pregnancy when we're putting a single euploid embryo. We need that next step, and I really think it's available.
So back to the very first question that one of the attendees asked, whether we can utilize the same technology to actually help us understand how the culture media is and conditions, growth conditions. And someone answered that question saying it's going to be expensive to do so. And that's a whole different discussion. But just looking at embryo grades, we know that is very subjective. And usually, you can ask 3 or 4 embryologists to score. They may score differently, but also it's not really reflective of the biology of the embryo. Morphology and the biology don't overlap 100%. There is certainly correlation there. Do you think, does Maldov has any space in assessing the metabolic activity of the embryo during the culture, the growth culture, where you could say those are new criteria of scoring the embryo?
Right. Look, if you have a Maldi in your lab, you can play with it. Running the test is really not expensive at all. The expense is the machine. The actual reagents that we use to run the assays are actually very affordable. Very affordable. You can look at anything you want. Obviously, you don't want to keep removing embryos from incubators and testing them. You definitely don't want to do that. But clearly, this spent media, most labs just throw the spent media. You go do a transfer and you just throw this away. I think this is something that you should freeze. And if you have a Maldi, you can look at all these things. Okay. Yeah. So stay tuned, Nabil. I really think we're in the infancy of what we can know from looking at spent media.
Yeah. But back to that same point. So you could, for example, if you know the profiling of AA grades, and you compare, you know, a large number, you may be able to find certain patterns that, you know, either they have certain peptide distribution or transcripts or large molecules, but you could use that new criteria to rescore those embryos instead of using the kind of the traditional.
But that's exactly what we've done. Yeah. We said, look, these, you know, even though most embryologists that are really good can grade, you know, using Gardner's criteria quite well, okay? All we're saying is this is possibly a more important way to grade embryos than just by looking at them.
Fantastic. Well, this is very exciting. I can't wait to see what's going on in the literature, and I'm tempted to kind of myself to look into this. Very nice presentation.
Thank you very much.
A lot of information. I really appreciate your inputs. I was excited when you decided to talk about this topic because, you know, I remember our conversation a few years ago. You were talking about the Maldov and the Maldi TOF and mass spec and metabolic activity. And a few years later, people just started to talk about it. And you've been working on this for a long time. And yes, some names are kind of pop-ups a little bit more in the field, but really you've done this virtually 4 or 5 years ago, right?
Yeah, we started working with the group that actually that is the most expert in actually running the Maldi. And they were running it on really refrigerators. This is how humongous these machines were. And I visited the lab in London and looked at these. They had 2 of them, absolute, you know, they're beasts. Now it is, you know, cutting edge. You see this, the new Maldi, it is so exciting. And it has come down substantially in price. So before, only very big research programs had a Maldi, and very few actually worked on Maldis. Now with, you know, with Shimadzu, and there's another company called Brooker that basically have made these Maldis much smaller. This is something you can put in your own lab.
Awesome. Very nice. Well, thank you so much. Thank you so much for the presentation, for the information, and I really appreciate your time. You taking time to present that to us.
My pleasure.
Yeah, thank you so much. Stay tuned for the next webinar, probably in the next week. And with this, thank you so much, and I'll see you soon.
Good night, everybody. Take care.
Bye-bye.
Bye-bye.